I think I can start. Okay. So today I'm going to introduce our project called GenPIP. Okay. So GenPIP is the in-memory acceleration of genome analysis via tight integration of the nanopore genome analysis steps. So first, I will give an overview of the whole project. Then I will go to the details. Genome analysis enables us to determine the order of the DNA sequence in an organism's genome. It plays an important role in the personalized medicine, outbreak tracing, understand the evolution, and so on. Modern genome sequencing machines extract the smaller randomized fragments of the original DNA sequence, which is known as reads. And this project will mainly focus on the Oxford nanopore technologies, which is known as ONT technologies, because it's a widely used sequencing technology, and also it provides portable sequencing devices. As you can see on the right side, it's just a palm size, and it has high throughput. Also, it's very cheap. So we observed, because there are multiple steps in genome analysis, and we observed there are two main limitations in the current nanopore genome analysis. So first, there's a large data movement between multiple steps. And second, there's a lot of wasted computation done on the data that is later to be discovered to be useless. That's why we propose GenPIP. GenPIP is a fast, energy-efficient in-memory acceleration system for the genome analysis pipeline via tight integration of genome analysis steps. GenPIP has two key techniques. The first one is the trunk-based pipeline, which provides a fine-grained cooperation of genome analysis steps. And the second one is early rejection, which timely stops the execution on useless data by predicting which reads will not be useful. GenPIP outperforms the state-of-the-art software and hardware solutions. So, okay, then I will go to the detail about this project. First, I will talk about the background and motivation of this project. So, first, I introduced the genome analysis pipeline. When we sequence the genome with the nanopore sequencing machine, we get the raw signals and we store them in the storage. And after that, we read out them to the to execute the first step, which is base calling. And during the base calling, we translate the raw signals into the bases, which is known as ACGT characters. And here you can see that we translate them in the granularity of a trunk, and then we assemble them into a read. Here, the green one represents a high quality translation, and the red one represents the low quality translation. After we finish all of the translation, we store the reads in the storage and continue with the next step, which is usually the read quality control. In the read quality control, we discard the low quality reads, and then we store the high quality reads in the storage and continue with the next step, which is usually read mapping. In the read mapping, we compare the reads to the reference genome. And if the reads cannot be mapped to the reference genome, then we discard it. If the read can be mapped to the reference genome, then we store the mapping results. So I go more details about these three key steps. So first step is base calling. Base calling usually uses the deep neural networks to ensure the base calling accuracy. So the input of base calling is the raw signals. And when we do the base calling, we translate the raw signals into bases and calculate the each base quality at the same time. And the output is the long reads along with the quality score for each base. So the second step is read quality control, from which we have the base quality scores for the long reads as the input. And we calculate the average quality score of this read. And then we compare the read quality score with the threshold. And if it's higher than the threshold, we define them as a high quality read. And if it's lower than the threshold, then it's low quality reads, and we discard the low quality reads. So the third one is read mapping. In the read mapping, we have the pre-analysis step called indexing, in which we reorganize the reference genome in the, for example, in the shape of a hash table, which enables us to query the reference genome faster. And for the read mapping, the input should be high quality long reads from the read quality control, and then we go through the seeding step, and in which we use the we generate the subsequence from the reads and query the hash table which stored the reference genome, and we get the possible locations of the subsequence inside the reads. Then we do the chaining, in which it identifies the candidate regions and output the chaining score. And finally, we execute the alignment step if there's a chain. So the output should be the mapping information here. In all of these steps, we found two main limitations. The first is large data movement. We use the human data set as example to explain in detail. So, for example, in this human data set, we have nearly 4,000 gigabytes raw signals. And after base calling, we still have 500 gigabyte reads. And after read quality control, we still have more than 400 gigabyte high quality reads. And after read mapping, we need to store the final result. So as you can see here, between each analysis steps, there are large data movements. So the next, the second limitation is the waste computation. We use the same data sets to explain more. So here in the example, after base calling, we have all of the reads, but after read quality control, we only maybe have less than 80% of high quality reads. And after read mapping, we only have less than 70% of the reads. So here we conclude that there's a considerable amount of computation on useless data due to the low quality reads and unmapped reads. I will explain more about the state of art work. So NVM-based PIM is an efficient technique to reduce the data movement by processing data using or near memory. And so here is an example that uses the NVM-based PIM for the vector matrix multiplication operations to accelerate the base calling, because it's the dominant operation inside the neural network applications, and base calling using the neural network to translate the raw signals to the reads. And here are more details about the structures. So basically in the RM array, you store the matrix in the RM cell, and using the multiply accumulation operation so basically you apply the voltage as an input vector, and then you collect the sensing the voltage on the bit line to get the result of the multiply accumulate operation. So here is an example to visualize the operation. So first of all, we have the resistance represent the vectors, and we apply the voltages on the watt line, and according to the Kirchhoff's law, and we accumulate all of the current on the bit line, and through this we do a vector multiply vector operation in a read cycle, which is really efficient, just with a sensing operation. So here is some more details that how we can do the vector multiply matrix operation. First of all, we store the matrix in the RM array, and then we convert the vector, input vector, and apply them on the watt line. And through the sensing, we can get the output. So as you can see, we can do a vector multiply matrix in just one read cycle. Here is overall like design for the neural network acceleration. So we basically have the crossbars, and which can do the vector multiply matrix operation, and we have other units such as the sigmoid operation and max pooling operation to support the other operations inside the neural network application. So with all of this design, you can use this kind of chip to accelerate the neural networks very efficiently inside the memory. So this is one accelerator prior work to the base designed for the neural network, which we can also use for the base calling because it's a neural network accelerator. And the second one, there's also some other accelerators for the read mapping, since it's also a very time-consuming step inside the whole pipeline. And here is an example that we use the NVM-based PIM for the search and addition operation. And since the search and addition are the dominant operation in the read mapping step. So these are two like representative accelerators in memory accelerators for base calling or read mapping. These kind of in-memory accelerators, they do reduce the data movement in a single genome analysis step. However, it exacerbates the data movement overhead between the analysis steps. And we conclude that there's no prior work tackles data movement between analysis steps and reduce the useless computation. So our goal is to efficiently accelerate the entire genome analysis pipeline while minimizing the data movement and the useless computation. We perform a study to quantify potential performance benefits, and the results are normalized to the performance of GPU. So firstly, we do the analysis on the NVM-based PIM accelerators for the separate base calling and read mapping step, which can achieve 2.7 times speed up over GPU. And second, we assume there's no data movement between these two accelerators, and which can achieve 6.1 times speed up. Third, we assume there's no data movement and no useless reads, which is ideal case, can achieve up to 9 times speed up over GPU. Next, I will introduce the GenPIP. GenPIP is the first holistic in-memory accelerator for the genome analysis pipeline, including base calling, read quality control, and read mapping steps. GenPIP has two key techniques, trunk-based pipeline and early rejection. So first, I will introduce trunk-based pipeline. It enables fine-grained pipelining of the genome analysis steps inside the nanopore sequence. And it processes reads at the trunk granularity, which usually is much smaller than a read. It's subsequence inside the read. So Trunk-based pipeline increases the parallelism by overlapping the execution of different steps at the trunk granularity. And CP also reduces intermediate data by computing on data as soon as data is generated. CP also provides opportunities for ER by analyzing a read at trunk granularity. So we use an example that does a read consist of 4 trunks, C1 to C4. In the conventional pipeline, we base call all of these 4 trunks, and after that, we assemble them into a read, and we do the quality control on the read, and we do the read mapping on the read. In our trunk-based pipeline, we first base call a trunk, and at the same time, we base call the second trunk. We do the quality control on the first trunk, and we do the partial read mapping computation on the first trunk. And after that, finish all of the trunks, and we assemble them, and we finish the rest computation of read mapping on the granularity or read. So here you can see we saved a lot of computational cycles. The second key technique of GenPIP is early rejection. Early rejection stops the execution on useless reads as early as possible by using a small number of trunks to predict the usefulness of a read. Early rejection predict and eliminate low quality and unmapped reads from the genome analysis pipeline as early as possible. So here is a flow of the early rejection. First, we base call a small number of trunks, and then we check the average quality score of these trunks. If it fails, then we stop analysis. If it passes, then we base call more trunks. And we mapped the base call trunks so far to the reference genome and check the mapping score. If it fails, then we stop analysis. If it passes, then we continue base call the remaining trunks, and we execute the remaining computation in read mapping. So as you can see here, early rejection includes the rejection based on the trunk granularity, the trunk quality scores, and also include the early rejection based on the trunk mapping scores. And I give more details about the early rejection based on the trunk quality scores. So here is the goal of the ERQ CQLs is to accurately estimate the quality of the entire read by checking the quality of a small number of sampled trunks. So here is an example. In this example, the left one is the low quality reads and the right one is the high quality reads. And we made 3 observations. The first one is the range of quality scores for the trunks extracted from the high quality reads is greatly higher than that from the low quality reads. And the second observation is that a single trunk's quality score is not enough to predict the read quality score because, as you can see here in the yellow one, there are many trunks whose quality score are larger than 7 here. And the third observation is that the consecutive trunks quality scores are usually close to each other. Which indicating that sampling consecutive trunks may not be representative enough to estimate the quality of entire reads. That's why we propose to sample a small number of non-consecutive trunks evenly in a read to predict the read quality. So for the ER-based trunk mapping, here we have you observe that mapping a small trunks provide too many possible mapping locations, and we sample a small number of consecutive trunks in a read, and then we merge them into a bigger trunk, and we map this trunk into the reference genome to predict whether the read can be mapped or not. So it's worth to mention that both CP and ER can be applied on different systems, such as CPU, GPU, and PIM. And we implement CP and ER using PIM, since PIM is more efficient to reduce the data movement between genome analysis steps. We also apply CP and ER on CPU and GPU baselines and observe speed up and energy savings. So let's go through maybe a little bit detail about the GenPIP implementation. So GenPIP has 3 modules: base calling module, read mapping module, and the GenPIP controller. First of all, we receive the raw signals from the sequencing machine and store them in the EDRAM, and then we forward the signal trunks to the in-memory base caller. And after we calculate the base call, the trunks, and we calculate the base quality score using our PIM-based trunk quality score calculation, and then we forward both the base call trunks and the trunk quality score to the GenPIP controller. And then after that, we forward the trunks to the read mapping module, and so that we can get the read mapping result. To enable the early rejection, we get the information from the the trunk quality score calculation. And there's a ER controller inside the GenPIP controller to compare the quality scores with the threshold. If there's early rejection, then we send the signals to the base calling module to end the current execution on the read. And we also get the information from the read mapping module for the trunk mapping scores. And if there's early rejection based on the trunk mapping score, then we send the early rejection signal to both modules to terminate the execution of the current read. So here are the new designs in this project, and it's worth to mention that we have a very efficient in-memory seeding module. Here we have the base call trunks from the GenPIP controller, and we store them in the EDRAM buffer and forward the trunk to the query string generator. The query string generator is a shifted register whose length is the 2 times of the trunk of a substring. Then we can shift one character each time and generate a substring from a trunk very easily. And after that, we send compare the substring with the keys that's stored in the RAM-based CAM, and get the address to fetch the values which are corresponding locations in the reference genome. And then we store the possible locations and forward to the mapping controller. So through all of these tightly integrating the genome analysis steps, we can reduce a lot of data movement, also eliminate the useless computation as much as we can. So, okay, then let's have a look at the evaluation. And we use several simulation technologies, and also there are more details about it. You can see, read our paper for more methodology. And for the baselines, we use a CPU, GPU, and optimistic integration of 2 PIM accelerators, which we assume there's no data movement between these steps. And we also assume there's no intermediate data, which causes no overhead. And we use 2 data sets, E. coli and human data set. Here are the results for the performance. So GenPIP has achieves 41 times speed up compared to CPU, and 8.4 times speed up of GPU, and 1.4 speed up of optimistic PIM. And both more results in the paper shows that both CP and ER are critical to the speed up. And for the energy efficiency, GenPIP provides 22.8 times energy saving compared to CPU, 20.8 times energy saving compared to GPU, and 1.37 times energy saving compared with the optimistic PIM. ER is there's more results show that ER is especially critical to the energy efficiency. And we also provide several sensitive analysis which we did offline, and we by this pre-analysis we can decide like how many trunks we use to do the early rejection based on the trunk quality scores, and how many trunks we use to do the early rejection based on the trunk mapping. So here are some more results. You can check it in the paper. Okay, so, and there are more details in the paper, and please feel free to check it. And if you have any question, you can send email to me. Yeah, so to conclude, the problem we're targeting in this project is that the genome analysis pipeline has a large data movement between genome analysis steps and a significant amount of wasted computation on useless data. And so our goal is to tightly integrate genome analysis steps to reduce the data movement between steps and eliminate the computation on useless data. So that's why we propose GenPIP, the first in-memory genome analysis accelerator that tightly integrates genome analysis steps. GenPIP has two key techniques, a trunk-based pipeline and a new early rejection technique. GenPIP outperforms the state-of-the-art software hardware solutions using CPU, GPU, and optimistic PIM. So that's all. Thank you for listening. Feel free to ask questions if you have any.